Okay, we left off on the first line of defense. We went over a bunch of first line of defense, like skin and gastric juice and things like that. So now we're on second line of defense, and now this is the pathogens in the body, in your interstitial fluid or bloodstream, something like that. So we have to treat it a little bit different. Our first line of defense didn't work. We got past the epithelial tissue, for example, like your skin or the lining of your esophagus or something like that. And so there's different ways to approach this, and these are the 4 types, and I've got a slide on each type. We've got some different cells that can work, some different proteins, fever and inflammation. So I've got a slide on each one of these. Okay. So we got 2 kinds of cells that are in this second line of defense. Remember, this is still general immunity. This is not specific immunity. So one is a phagocyte. And there's slight differences, but really we use the terms phagocyte and macrophage interchangeably. So macrophages, phagocytes, we even use it as a verb. Like when we talked about blood, we said that the whole red blood cells are phagocytized by the liver and the spleen. That meant that the phagocytes eat it. So there's cells that eat other things. So in this case, the phagocytes eat pathogens. So some of them are hanging out, for example, in your lamina propria, like the tissue right under your epithelial tissue. You're going to have some of these phagocytes just hanging out there. So when you breathe in like I don't know, whatever, eat something you shouldn't have eaten, and that pathogen gets past your epithelial tissue, there's usually phagocytes that are fixed, that are sitting there, but some phagocytes are wandering around as well. They're just picking off pathogens. Another type is a natural killer cell. These are kind of like T cells once we start talking about T cells or T8 cells. Natural killer cells, they don't eat pathogens. They use these 2 weapons that you see here, perforin and granzymes. Perforin is like perforated paper, you know, it's got like those holes in it. So it makes perforations in the pathogen, punches holes in it. And it causes all this stuff to leak out of the pathogen. So that's cytolysis, cytolysis, cell breaking. So that's one tool it has. It has this chemical that it releases called perforin that causes cytolysis, and then granzymes. So here in granzymes I have apoptosis. Apoptosis is like cell suicide. So as far as our understanding is, cells have like this time built into them. They've got like this predetermined time that they're going to die. Granzymes push this date up. So granzymes tell the pathogens, oh, you know your date that you're supposed to kill yourself is today. Being very general here, but this is what they do. The natural killer cells induce what's called apoptosis. That's cell death.
Is it cell there?
Yeah, it's like, you can imagine it's like cell suicide. Cells have time, they've got pre-built times in their genetics, and they know it's just time to go with that. So granzymes, natural killer cells release these granzymes, and they're like, oh yeah, that time is today. So those are cells that are involved in the second line of defense. There are proteins involved in the second line of defense. And I put a few of them on here, and one that you might come across are called interferons. They use them in, okay, so first I guess we should talk about how viruses work. Viruses are what are called endogenous. I don't know if you guys can see the board on the recording, but maybe if you go to like a, I'm just gonna write the words and you go to like go to the upper right and you'll see like the picture, like my picture, and then you can like make it bigger just temporarily, but here's the words. You have exogenous and endogenous. And under endogenous, I'm putting viruses. And that just means, do you attack the cell from the outside, or do you attack the cells from the inside? So viruses work a little bit different in that they attack cells from the inside. First they get in the cell, then they start doing their damage.
So the first thing it takes cells from the inside.
Yeah, it's like a Trojan horse. All viruses are. They're so simple. They're a strand of DNA or a strand of RNA, and then they're covered by some kind of like protein. That's it. Like they're not even cells. They don't have all the stuff that cells have in them, like Golgi apparatus and ribosomes and stuff. So, like there's a debate on whether viruses are actually living or not, and I kind of evenly divided debate. But here's a cell, and here's like a nucleus in the middle. So the virus, what it'll do is it'll come up to the cell, and it'll shoot its DNA inside of the cell. And the DNA will go and hijack our DNA. So like viruses have instructions on how to make more viruses. So they get our cells to make more viruses. And the problem with viruses is that we really don't know we've been infected until it's in us, in our cells. And then once it's in our cells, this is going to piss me off. Once it's in our cells, then it's often too late. So what interferon does is that when certain cells called antigen presenting cells, I mean, it's okay, we'll talk about that later, but when certain cells are infected, they release a chemical, interferons, that go to other cells nearby and tell them that they've been infected. Because your body is able to fight off viruses if it only knew that there was a problem. And there are things that we can do. They're using interferons for other things like autoimmune diseases. They're trying that out. So you might see it being used for pulmonary fibrosis or something like that, because they're just trying it for some reason. And then transferrin, you've heard of that before. It moves around iron, right? So here it's got It works with like, it helps with immunity. Not immunity, it helps with our immune system. So some bacteria use iron to grow. Some pathogenic bacteria use iron to grow. If you can grab that bacteria, and if you can grab the iron and keep it away from them, then you can prevent their replication. That's something similar, like there's something similar in breast milk, lactoferrin it's released, and it kind of does the same type of thing. And then complement, that enhances antibodies, which we haven't even talked about, and it enhances phagocytosis. It's actually like a pathway, but we're just not going to talk about it. So there's 3 types of proteins that all work. So that's another thing we can use against pathogens. And then inflammation. So that's what inflammation is at the top. Actually, I've got both here, inflammation and fever. So inflammation is vasodilation and increased permeability. Increased permeability is filtration, pretty much. So it's like increased dilation, increased vasodilation, and then increased filtration. So you want to get certain things to that site. I was using the example the other day of getting stung by a bee. So your hand would swell. That's a reaction. You want to lock in. Swelling will kind of lock in that toxin from the bee sting, and it will get white blood cells and other things, get them to that site faster. So this is their fix. And then I have some chemicals, some chemotaxis. Well, these are some chemicals. They're kinins. So anyway, just so that I've written it, histamines and kinins cause vasodilation. They all kind of work together. Then you have prostaglandins. I'm going to come back to this. Prostaglandins intensify it. And then leukotrienes increase permeability. Leukotrienes increase permeability, and then they act as a chemotaxis agent, and a chemotaxis agent is like an attractant. I have it in another slide. Chemotaxis it brings the pathogen together with one. So write these down. They often work together. So, when you're like an antihistamine, right? Your like nose is running and like your eyes are kind of watery, and this is why, because you're starting this process out in your face, in your what's the word? In your sinuses, which are pretty much just holes in your skull, right? They get infected, and then this is one of the reactions. So that first bullet point is inflammation. I put fever, instead of making it in a separate slide, I put it as the second bullet point there.
So the vasodilation and increased permeability of blood vessels is inflammation, you're saying?
Yes. So you want to be able to move things out of the blood vessels and into the interstitial fluid. You want to be able to do that better. And you want the blood to get around better too, so you dilate your blood vessels in that particular area. Fever just simply elevates your body temperature. I mean, just the fact that your body is, trying to fight something, that is going to increase your metabolism, and that's going to increase your body temperature. So that's kind of a natural thing that happens. I'm not saying fevers are like great, but that just happens because your body's like doing stuff. It's fighting stuff. So microbes don't like it when your body temperature is 38 degrees. It should be 37. Or in Fahrenheit, whatever, 98.6 or something, 99. It should be around that. It should be around 99. Bacteria, viruses, they don't like 100 degrees. They don't like 101. It's too high. They don't grow. They've got particular temperatures that they like, and that's your temperature. They don't grow well in other temperatures. But what we tend to do is we tend to try to treat the fever, which, I mean, I get it. We should, I guess. I do. I shouldn't. But sometimes we should leave it. If it's 100, if it's 101, 100, we should just leave that alone. Especially in kids. If they're running 101, just leave it alone. Right? They usually tell you wait until it's like at least 102 or 102 and a half before you do anything. But that's what we do. We got to treat them. I came up with an invention once that I never used. I never did it. I never went through with it. But it was like one of those, it was like a Band-Aid, but it was made out of cloth. And it had a little chip in it, a little radio frequency chip. And it monitored your baby's temperature. You just put it on the forehead like a Band-Aid. And you can leave that baby in bed. And then it communicates with your phone. So you could like sit on the couch and watch TV. And you don't have to sleep next to your feverish child. And if it hits like a certain temperature, it'll just notify your phone. So I thought anything that allows us to be like worse parents would sell well. I would buy it. I would definitely buy it. I definitely don't want to, but I can't sleep at night because I feel like, well, now I don't give a shit at now. I mean, I really do. My kids are old. I'm not taking them to the hospital unless they're gone. Right, and even then, I might make the older one drive the younger one. So now they're teenagers, I don't care. But even when they were younger, like the first one, the second one I didn't care. If he got a fever, well, he's on his own. Hope he doesn't die. But the first one, it's like you lay next to them and you can't sleep because you think maybe while you're asleep, their fever's spiking. So wouldn't it have been nice to have something that did the job for you? You could just watch TV in your bed. Go on your phone and you don't have to think about your kids. That would have been nice. I don't know why I never jumped on it. Chemotaxis is like when a chemical stimulates this response. So not everything's gonna like stimulate the response. That's not an important slide. I don't know why I'm on there. All right, so all of that, we go back to it. The second line of defense and the first line of defense all fall under general immunity. They don't involve like T cells or B cells. It doesn't involve like what you're getting the vaccine for. None of this has anything to do with that. Like the vaccines don't make this better or worse. Vaccine's all about specific immunity. This is all general immunity. So this works for anything. Just only a certain level. Second line of defense works on any pathogen, but not when you got the Rona and there's like millions and millions of them by the end of day 2 in your body. Then this doesn't work. This is not enough. But this works with little things that you get day in and day out. And actually, you know, when you get like like allergies, like you're breathing in pollen, and that actually, now you kind of start, you know, if you feel like you're kind of like having a flu, that's specific immunity. Getting your immune system all activated for nothing. Because the pollen aren't, or whatever it is, ragweed, I don't know what it is. I just hear the word ragweed, and that sounds like something. Yeah, that pollen's not reproducing in your body, but we think it is. But second line, first line of defense, that's all general immunity. Just everyday stuff. Specific immunity is more serious. So you see I've written about a few different types of cells up here. You'll see on the second bullet point, there's T8 cells. On the fourth bullet point, there's T4 cells. And then you see B cells. So there's 3 types of cells. Let's see if I got a picture of it. I'll come back to this. So it's a 2-pronged attack, right? All right, so it's going to involve 2 types of cells, 3 types of cells. I don't think me touching it is making it. I don't think so.
It might be on the time plan where it goes out.
It's when they came in the other day to mess with it.
Oh.
And like people watching the recording, they don't even know what I'm talking about. Yeah, they're gonna have to fix this. So B cells, T cells, they're all made in the red bone marrow. Just like your red blood cells, just like all your blood cells. T cells and B cells are forms of lymphocytes. Remember, that was one of your 5 types of white blood cells. Right, so they're all made in the red bone marrow. B cells are going to stay in the red bone marrow. That's where they learn to become immunocompetent. T cells become immunocompetent in the thymus, which is sitting on top of your heart. So there's a two-pronged attack. So you see on the right side there's something going on, and on the left side there's something going on. And so on the left, there's different names for this, but we're going to go with the book here. And so the left side is called the cell-mediated response. Cells are the ones that are doing the damage to the pathogen. So on the left side, it's cells. It's T cells. They're directly confronting the pathogen. On the right side, you see B cells. That's called an antibody-mediated immune response. Antibodies were not actually cells. If you follow it all the way to the bottom, these red things, those are just proteins. And they're not actually attacking the pathogen. They're just doing stuff to the pathogen to slow it down. It's just like in war, you have the soldiers that actually have the guns and like shoot people, but then you got all the other people that are like support, intelligence operations and psych ops, whatever, I don't know. I just like saying psych ops. But they're not actually killing people, but they're doing other things. And then in the middle here, there's a third type of cell called a T4 cell. And that's kind of like a command and control center. I'm gonna move back one slide. This is the same thing. So, two-pronged attack consisting of cell-mediated and antibody-mediated immunity. They need each other. The cell-mediated, they'll get overwhelmed in the fight. They won't be able to. They get overwhelmed. But antibodies by themselves can't kill off a virus. Antibodies, they make it easier to kill, but they can't do it by themselves. Because antibodies they're just proteins. So cell-mediated immunity is going to involve T8 cells. There's different names for this. We're just going to call them T8, but they're also called like CD8 or killer T cells or something like that. Right? So these T8 cells are eventually going to clone and they're going to become cytotoxic T cells. So we're going to talk about the process by which that happens. The antibody side, this right side here, involves B cells. B cells are going to, when they get stimulated in the right way, they're going to become plasma cells. Plasma cells are going to secrete antibodies. We're going to talk about that. So that's what I have on. So antibody-mediated immunity involves B cells, which are going to clone to make a bunch of plasma cells, and plasma cells secrete antibodies. Helper T cells, or T4 cells, or CD4 cells, however you want to say it. They call them, often they'll call them helper T cells. They mediate in both processes. So without the helper T cells, you won't have either side happen. So let's talk about antigens first. Antigens are the pieces of a pathogen. Are any of you guys cold, or is it just me?
It's freezing in here.
What is going on in here?
Yeah.
It's cold outside.
Yeah.
Run that thing for.
Keep the germs down.
Yeah, I think my baby goes in.
Man, my fingers are just. This is so cold here all the time. Why do I feel like that? Oh, that's a huge problem. When I bring in like the Wendy's coffee, it I only get about 5 or 10 minutes out of it. Yeah. Yeah, so antigens are just a piece of the protein. A piece of the, actually, or it's a piece of the membrane, or a piece of the thing that's around the pathogen. So like a bacteria's got like this protein coat, and so do viruses. And then they've got these proteins that are stuck to them. And so that's what an antigen is. You don't actually have to look at the entire protein. You don't have to look at the entire bacteria. You can just look at a piece of the bacteria, which is the antigen. But antigens have to provoke the immune response. Some antigens don't do it. And so that's immunogenicity. So sometimes an antigen won't be sufficient to get your immune system all jacked up and ready to go. And then it has to react. So the cell has to react with the pathogen has to be able to react with the antibody. Or the cell, or the T cell. And some pathogens are really good at that's like what they'll do is, for example, some pathogens will, as they're coming, like viruses, when they come out of our cells, when they exit our cells, they take some of our cell membrane and they wrap themselves with it. So they look like us.
so we don't recognize them. So that's not going to have any immunogenicity. Or what viruses will do is they'll provoke our immune system, they'll have immunogenicity, but they won't have any reactivity. Because what they do is like they provoke our immune system and then they change their antigens. It's like changing the flag on your uniform. So, we get ready to go fight a certain type of virus, and our immune system gets out there and go kick some butt, and they're like, what virus? That's like, oh no, they're gone. They left. And then we got to go and start everything from the beginning. The immune system can't be like, oh, well, I'm here to kick some ass, so I'm just going to kick your ass. It goes back to the base, and it's got to retrain, learn on a new virus, and they retrain and retool up, and you feel like crap for a couple days. They go back out to fight the good fight again, and the virus has changed again. They're like, what? They're not here anymore. Just us. And epitopes, they're like the little pieces of the antigen. Actually, you don't even need the whole antigen. You only need like little pieces. So we have these things called MHC molecules, major histocompatibility antigens. So on cells like our blood cells are like, type A and type B or type O. We kind of learned about that. But the rest of our cells have very specific antigens, not type A or type B. They're specific just to you. And so all of our cells, pretty much all of our cells, have these MHC1 molecules. So they're built into our cells. And there's like pieces of them that are sticking out on the outside. And so I view it like a name tag. Like a name tag saying class, right? And if you have a different name tag, you come to the party with a different name tag, you're gonna get cut. Shame. No, they're just gonna shoot you. You're dead. You come to the party, your choices are no name tag or have a name tag that says Klaus. But you better not have one that says like Scott, because that's not happening. No, it says Scott. Chip. I just think any guy at a party named Chip is just good. So except for our red blood cells, right? Our red blood cells are different. They've got the A or B, the Rh positive, negative, whatever. They got like a little bit of a different system going on, right? But the rest of our cells use these MHC. All right. So we have certain types of cells in our body called antigen presenting cells. Because the thing is that T cells are like the warriors of our body, but they're not very smart. They're good at killing things, but they don't know what to kill. You have to go and show them exactly what to kill. They're not even going to clone and make their clone army until you show T cells what it is that they have to go kill. And that's what antigen presenting cells do. They take pieces of the bacteria or virus and they present it to the T cells. Now I'll show you how they work in the next slide. So they have also MHC molecules, but they're like a little bit different. They're not embedded in the plasma membrane. They're like just sitting on the surface. But point is, where's my picture? There it is. But what's this slide? All right, we'll get to that. All right, this is an antigen presenting cell. This whole thing, it's not like a round cell. It looks like an oval or whatever, like a blob.
So this whole thing is the antigen presenting cell. Here's the antigen on the left side. So what the antigen presenting cell does is that it phagocytizes the antigen. So there's, or I'm sorry, the pathogen. It phagocytizes the pathogen. Here's the, pretend this is a bacteria, and the maroon colored square and triangle and rectangle, those are like antigens, right? So it's eating it. Here at number one, it's phagocytizing, it's eating it, and here it is, it's taking it inside. And it rips it apart. So it's ripped apart the bacteria. These shapes, the square and the rectangle and stuff, that's the antigen. So at the same time, it's making new MHC molecules. It's making its own name tag. But what it's doing is that it's incorporating pieces of the bacteria into its name tag. And so that it makes the MHC molecule, but it's got pieces of the antigen. It's got antigens from the bacteria on it. So now something doesn't look right. It's like when you see it's like you're on the interstate, and then you hear behind you, and it like passes you, and it's like so loud, and it's got like a bunch of neon lights, and it's got like this really high spoiler on the back. You take like another look at it. You just look at it twice. You're like, wait a minute. Wait, that's a Dodge Neon. You're not fooling anyone. Like something doesn't look right enough. So that's what it's like to the T cells. So this antigen presenting cell is going to go on to the army base, and it's going to go walk into the bar where all the soldiers are hanging out, and it's going to walk around the tables and say, "Hey, look at this thing on me. Notice anything different? Looks a little bit weird."
It's like the other side, big thing.
Yeah.
Okay.
I mean, not the Dodge Neon part, but
Oh, yeah.
and you don't have to memorize this. Essentially, what's happening is that it's eating. This is what antigen presenting cells do. They eat the pathogen, like the bacteria. They eat the bacteria. They rip it apart, and they use its pieces to make its MHC molecule. That's it. It's going to go to the T cell. It's going to walk up to a bunch of T cells, and most of the T cells aren't going to care, but a few of those T cells are going to recognize that something's wrong. I think that's good, right? A few of the T cells are going to recognize that antigen. And so now they're getting all hyped up, right? A few of the T cells recognize it, and they're telling the other ones, are like, "Hey, this is like an attack. You know, what are we going to do?" And they're like hitting each other in the chest, and they're like getting ready to go, right? They're all pumped up. They've been drinking too, so they got a lot of confidence. And so, so here they are. So these T8 cells are getting all excited because the antigen presenting cells just presented the antigen to them. And now they look over at the T4 cell and they're like, "Hey, can we go? Like, we want to start cloning. Can we start cloning?" And then the T4 cell will tell it, "Yes, you can start cloning." That's when they start to clone. So you need two types of stimulation.
the first stimulation comes from the antigen presenting cell. The second stimulation comes from the T4 cell. Let me see if I have this written down anymore. It's called interleukin 2. Oh yeah, I've got it written down. Oh, right here. I got the whole thing written down here. Why do I have all this? I forget what other slides I put in. All right. I'll go to the next slide in a second. Here's the I'm trying to follow this myself. All right, here's the here's like the T8 cell. Oh, you know what? Right. So T8 cells are normally inactive until they bind to an antigen. The antigen comes from the antigen presenting cell. That's the first stimulation, that second bullet point. That's the first stimulation. The second stimulation is they get a chemical called interleukin 2 from the T4 cells. Or helper T cells. Now the T cells, T4 cells can clone to cytotoxic T cells. This is why HIV is like really difficult, because that's like the second time I talked about it, but HIV specializes, that virus specializes in attacking helper T cells. So if you attack the helper T cells, you've cut off both pathways. If you don't have helper T cells, you can't send that signal to the T8 cell that it's okay to go, that it's okay to attack. And if you notice on the other side, I think it just waits for a few seconds. Now it's not doing it at all. All right, that worked well. If you guys, there's another TV up there. I'm almost done. So. I don't know why it's not working. Oh. All right. Yeah, so when the T4 cell gets infected, you see it's really sending, you know how I was saying that it sends out interleukin 2? It does it to both sides. It's sending it to the B cells and the T cells. We've only been talking about the T cells, or the T8 cells. So when it gets both stimulations, then it will start to clone and become cytotoxic T cells. So you get millions and millions of cytotoxic T cells. These cytotoxic T cells are going to use the same tools as natural killer cells. Let me make sure I don't have that. No, I don't. All right, so how do killer T cells, well, so I use these different words. I'll use cytotoxic T cells. So T8 cells, once they get the stimulation, they start cloning into cytotoxic T cells. Cytotoxic T cells use perforin and granzymes. Perforin and granzymes, just like natural killer cells, same thing. They've got the same weapons, it's just that cytotoxic T cells only kill one specific thing, but a whole bunch of them. They kill millions of that one specific thing. Natural killer cells can kill lots of things, but only a few of them. But they use the same weapons, perforin and granzymes. that's the cell-mediated immunity side. There's also an antibody-mediated immunity side, so there's antibodies too. And so it's the same idea. You have a B cell, and the B cell, you have to activate the B cell. So antigen-presenting cells are going to activate the B cell. They're going to get all anxious, and they want to start turning into plasma cells, and they're waiting just for the helper T cell to tell it, okay, go ahead. So that's like the helper T cell. So the helper T cell gives it the permission to go ahead and start becoming plasma cells, and they start becoming a whole bunch of plasma cells. So B cells become plasma cells. Plasma cells are going to secrete antibodies. So this is like the process here, right? So the B cell, gets the first stimulation from MHC molecule. Then it gets the co-stimulation from the helper T cells. And then they become plasma cells that secrete antibodies. We're going to talk about what antibodies do. Got one more slide. Yeah, this is the other side. This is the antibody-mediated immunity. So whenever your immune system's getting activated, you're doing kind of both of these things. You have a cell-mediated response and you have an antibody-mediated response. And that's really why you're feeling like all achy and sick and stuff, because your immune system's really doing a lot of work. It's trying to build this army. So antibodies are not actually cells, they're just proteins. Pieces of proteins made by cells. Yeah, I need to mess with it again. This is what antibodies do. This thing that you guys fix, it keeps like every couple minutes it goes orange. It goes like for like 15, 20 seconds, and then it comes back, and then it goes, and it comes back. And I don't think me touching it is making it come back. I don't know.
Okay.
It's not like because I'm not interacting with it. I don't know.
Only that one doing
Yeah,
it?
this one's fine.
This one's fine. That's something I'll have to report to them. Hopefully.
See if we can all solve the problem. So this is what antibodies do. So they block. Okay, so bacteria, the reason the bacteria make you sick is because of toxins. Bacteria eat and they poop, just like we do. And that's what the toxins are. They're eating stuff in our body, and then that's the byproduct. It's the byproduct that makes us sick. So if you can block that, it kind of makes us feel a lot better. And then preventing viral attachment. Remember, viruses will come up to a cell and insert their DNA or RNA. They'll insert their genetic material into our cells. And they get our cells to make them. We actually make the viruses. That's what's so messed up. They use our cells to make them. So if you can keep them from attaching, you think it's because, if you can keep them from attaching,
I'm thinking they have it on, like after so many minutes of not being.
now I feel like when I move this, it comes back on. Maybe I'll try that. Maybe it's just a computer setting.
It is.
I'm gonna try that again next time it goes off. And if it's if my mouse gets it back on, then it's not. It's the computer setting. So yeah, if you can prevent the viruses from attaching, then they're not gonna infect you. And then immobilized bacteria, cilia and flagella, not so much flagella, especially cilia. Bacteria move around with cilia. The little hairs that they use to, you know, it's hard. It's hard to get them. And then agglutinizing means that you grab a bunch of them and just hold them together. It's like to go back to talking about kids, like when you're at Chuck E. Cheese and it's time to go, and you like grab one kid, and then you got that one squirmy kid, and then you're trying to get another kid, but when you try to grab that other kid, that first one runs off, and then you got two more running by you, and you can't get them all together, right? So what if there was something that would come and like chop off their legs and just grab 5 or 6 of them together? That'd be nice and easy to get all the kids. When my kids were young, Chuck E. Cheese and McDonald's. Thank God for McDonald's playground. Get my computer, study, sit by the door. I don't have to look at my kids for hours. Just climb on those things and get to cold.
What they're planning to do, what their goal is.
When they won, you could buy more chicken nuggets. And Chuck E. Cheese is the other good place. You sit up front by the door and you let your kids be someone else's problem for hours. They're in some other kid's birthday party. They're learning to gamble. So glutinizing antigens, you gathering them together, and then if you look at the shape, if you were to look at the shape of an antibody, it's got like a flag on it. It's got like this stem, like a flag that sticks up. So whenever they're walking around, you can see them like in a crowd. Right, so phagocytes can come in and start eating these as well. It makes it, they could join in the party. So antibodies aren't killing pathogens, they're just making them a lot easier to kill. Now see, I'm doing this with my mouse and nothing. No, I don't think it's related. I don't know. That's my last slide. Hold on, let me just, that's my last slide. That's all we're going to do today. That's kind of a lot to wrap your head around. So we've got sum it all up. To sum it all up, if I can. Got immunity. Immunity, by the way, self versus non-self. That's how your immune system sees things. You've got general and specific. Under general, you have 2 types of general. You have the first line of defense, and you have the second line of defense. First line of defense, the difference between the two is that it did not get past the epithelial tissue, it did get past the epithelial tissue. So if you swallow, if you eat like old food with bacteria in it and you swallow it and it goes into your stomach, and the bacteria gets killed by your stomach acid, and then you poo it out the other end, that's all first line. That never got in your body. Like technically, it's not in your body.
It just went through a tube and out the other end. If you eat that food and while you're chewing on it, that little bacteria lodges into your teeth and then gets into your gums and into your bloodstream down that second line, right? So that's the difference. Then under first line, we had things like skin and vomiting. I had to spell emphasis. And gastric juice, hair, yeah. And then under second line of defense, we had the cells, we had the antimicrobial proteins, fever, and inflammation. So that's general. Under specific, it's one reaction, but it's kind of broken into two pathways. There's the cell-mediated, and then there's the antibody-mediated. So getting some bacteria in your food that might give you a little bit of diarrhea, or maybe you throw up once, but then that's it. It's kind of over. That's general. Getting like the flu, when that thing gets in your body, it starts going. Like it's making millions and millions and millions of viruses. You know, it's not just a little bit. So that's going to be specific immunity. And by the way, I never even mentioned it, but some of those T cells, some of those helper T cells, some of those B cells, they're called memory. Like after the battle's over, they're like the veterans, right? They stick around and they remember. So if Nazis ever come back, they're like, oh yeah, we know who that is. So, that's all I need. Provoke your immune system. Some of them stay around and remember, so that when you get infected again, your body just takes them out right away. As soon as the first plane load of Nazis comes in from Germany, it lands on the tarmac, gone. They're going to Delta Force them. They're finished. Delta Force. They're going to check. So cell-mediated, antibody-mediated. Cell-mediated is going to involve the T8 cells. Antibody-mediated is going to involve B cells. And then the command and control center is the T4 cells. They're going to get the stimulation to both sides. Right. T8 cells are going to clone, and they're going to become cytotoxic. Cytotoxic T cells. B cells are going to become plasma cells. Now, plasma cells are going to secrete antibodies. That's kind of a flow chart of our immune system. It's just kind of general, it depends. Did it get into your body or not? But here, once specific goes, this whole thing's going to go, right? You're going to have cell-mediated and antibody-mediated. They don't pick one or the other. All right, that's a lot of stuff. So any questions?